I am Dr. Shri Devi, Associate Professor of Botany, Shri College for Women, Autonomous Salem 16. First of all, let me thank our benevolent management and our principal for giving me permission to develop this e-content video. Today, we are going to discuss about the properties of enzymes. First of all, let me tell you what are enzymes. Enzymes are the substances which can speed up the rate of biochemical reaction in a forward direction. Hence, they are termed as biocatalyst or catalyst of life. Normally, a catalyst is a substance which increases the speed of chemical reaction in a forward direction. At the same time, it does not undergo any change even at the end of the reaction. The same thing happens to the enzymes also, but the enzymes are synthesized by the living cells and they are catalytic in function, hence they have been termed as biocatalysts. Enzymes are made up of proteins. So proteins are very sensitive to heat. So enzymes are also known as thermolabile or thermolabile in nature, and they are very specific in their action. Enzymes can act upon only a specific substrate. It cannot change its substrate. If we changes the substrate, what happens? The enzyme will not act on the specific substrate. And enzymes are colloidal in nature. They are not true solutions. At the same time, they are colloidal in nature. Colloids do not have any filterability. Enzymes are neither true solution nor suspension. They are colloids. In recent years, certain non-protein enzymes have also been identified. Next, we get into the topic properties of enzymes. The properties of enzymes are categorized into 5 different heads based on the different property. Number 1, catalytic property. 2, reversibility. 3, substrate specificity. 4, heat sensitivity. And 5th one is pH sensitivity. First one, catalytic property. As we discussed earlier, the enzymes are catalytic in nature and they have an extraordinary catalytic activity, because they are required in a very small quantity to carry out the reaction in a larger quantity of substrate. So a large quantity of substrates can be easily convertible into their products with the help of a small amount of enzymes. But at the same time, the enzymes remains unchanged after the reaction. The turnover number of enzymes are very particular and it is most important, because this turnover number decides how much amount of enzymes or how much quantity of enzyme is required to carry out the reaction. The number of substrate molecules which have to be converted by a molecule of enzyme, how many molecule of enzyme per second, is known as turnover number. This turnover number of an enzyme is calculated, based on which the quantity of enzyme can be calculated. The active site is normally saturated with the substrate molecule. Only then we can calculate the turnover number. There are number of enzymes which have a specific turnover number. Here I can give some 6 different enzymes and their turnover numbers. Carbonic anhydrase has a turnover number of 6 lakhs. Acetylcholine esterase have 2,80,000 turnover number. penicillinase 2000, chymotrypsin 100, DNA polymerase 15 and lysozyme 0.5. The second property of the enzyme, as I told you, is specificity. And enzymes are very specific in their action, which means they act only on the specific substrate. For example, lactase will act upon lactose and cellulase will act on cellulose. Proteinase will act upon proteins. Likewise, it goes on. Enzymes are very particular in its reaction also. Say for example, oxidation is carried out by oxidases and reduction by reductases. And there are a group of enzymes called oxidoreductases which can carry out both oxidation and reduction reactions. And enzymes, in some cases, this kind of specificity may not be too strong. Exceptions are always there. And different enzymes show different types of specificity, and the specificity can be categorized based on the type. Say for example, it may be bond specific or group specific, substrate specific, stereoscopic specificity, geometric and cofactor specificity. For example, if one particular cofactor is not available, the enzyme will not act or it remains inactive. Only when that particular cofactor is available, then only the enzyme will be active. Third property is reversibility. Most of the enzyme catalyzed reactions are reversible. It may be occur in forward direction as well as the reverse direction or backward direction. The reversibility of the reaction is completely depends upon the requirements of the cell. If there is no product or a very little product, the biochemical reaction can take place in a forward direction, which means the reactants are converted into products. Say for example, glucose 1-phosphate is converted into glucose 6-phosphate in the presence of an enzyme called phosphoglucomutase. When more glucose 6-phosphate is formed, what happens? The same phosphoglucomutase can convert glucose 6-phosphate into glucose 1-phosphate. So this is purely depends upon the requirement of the cell. But in certain cases, there are separate enzymes available to carry out the reaction in forward and backward directions. And some of the enzyme catalyzed reactions are also irreversible, which means they are not reversible. They occur only in a forward direction. Right? The fourth property is heat sensitivity. As we know very well, enzymes are made up of proteins and proteins are very sensitive to heat. So in turn, enzymes are also very sensitive to heat as well as the temperature. That's why the enzymes are called thermolabile. The maximum activity of the enzyme will be at its optimum temperature or normal temperature. The right temperature at which the activity of the enzyme is maximum, we call it as optimum temperature. So what happens when the enzymes are subjected to a low temperature or higher temperature? When the enzymes are found in the lower temperature, it remains inactive. It does not participate in any kind of reaction due to very low temperature. This is the reason why we preserve all our vegetables, natural foods, fruits in a refrigerator to preserve. Because if you reduce the activity of the enzyme or if the activity of the enzyme is nil, the shelf life of the fruits and vegetables would be higher. And the enzyme activity increases with increase in temperature up to a certain level. When the enzymes are exposed to a higher temperature, probably 60 to 70 degrees centigrade, the enzymes will be destroyed or denatured. The destroyed enzyme or denatured enzyme will not show any activity. That's why the enzymes are said to be heat sensitive. The fifth property of an enzyme is pH sensitivity. Like temperature, enzymes are also very specific in their pH requirement. What is this pH? pH is defined as the negative logarithm of hydrogen ion concentration. So when the enzymes are exposed to a very high pH or a very low pH, beyond its optimal pH, what happens to its activity? Its activity is reduced, or it does not take part in the reaction, because pH is one of the essential factor which influences the activity of the enzymes. Hence, some of the enzymes are active in acidic pH and some of them may be active in alkaline or basic pH, and very few are active only in neutral pH. So the right pH of the enzyme is denoted as optimum pH. As you know very well, proteins are also having optimum pH. Since enzymes are made up of proteins, they do show the same character of a protein. So these enzymes are also having an optimal pH. So the enzymes show the maximum activity only in its optimum pH. When there is any change in its pH, definitely their activity is highly reduced. In this slide, I'll show you some of the enzymes and their substrates, their optimum pH and its location. So pepsin, it's an enzyme used for digestion. It is synthesized in human stomach. It will act on the peptide bond, and the optimum pH for this enzyme is around 1.5 to 2. The second enzyme is sucrase. Sucrase will act upon the substrate called sucrose. Hope you know this is a disaccharide, and the optimum pH of the sucrase is 6.2. This is also acidic pH. And this sucrase enzyme is synthesized in the small intestine. The third enzyme is amylase. And you know very well, amylase will act upon the substrate called amylose. And the pH of amylase ranges from 6.7 to 7. This is acidic to neutral pH. And the location of this enzyme is pancreas. The next comes urease. Urease will act upon urea, and it is secreted in the liver, and this will act only in the neutral pH, that is 7. The next enzyme is trypsin, which will act on the peptide bonds, which will be helpful to break up the peptide bonds, and this is synthesized in the small intestine. The optimal pH of trypsin ranges from 7.7 to 8. Next comes lipase. Lipase is an enzyme which acts upon the complex molecule, biomolecule called lipids. The lipase will act on the lipid, and the pH requirement of lipase is 8. This is an alkaline pH. And the location of this enzyme is pancreas. So, we have discussed the properties of enzymes in various heads. So, enzymes are very specific in substrate level as well as temperature, pH and its activity. So, they are very particular. If you alter the pH or alter the temperature, what happens? The enzyme activity remains unchanged, or the enzymatic activity does not happen. it does not react and all, or it does not proceed the reaction in a forward direction. They may be broken down, or they do not involved in the reaction. That's all. Thank you.